Hello, thank you, Ted. Good afternoon, everyone. My name is Helen Talley McRae, and I work in the One Health Office of the National Center for Emerging and Zoonotic Infectious Diseases at the Centers for Disease Control and Prevention. On behalf of the One Health Office, I'm pleased to welcome you to the monthly Zoonoses and One Health Updates call on June the 5th, 2019. ZOHU call content is directed to epidemiologists, laboratorians, scientists, physicians, nurses, veterinarians, animal health officials, and other public health professionals at the federal, state, and local levels. Please be aware that CDC has no control over who participates on this conference call. Therefore, please exercise discretion on sensitive content and material, as confidentiality during these calls cannot be guaranteed. Today's call is being recorded, so if you have any objections, you may disconnect. Detailed instructions for obtaining free continuing education are available on our website and will be given at the end of this call. These presentations will not include any discussion of the unlabeled use of a product or a product under investigational use. The Planning Committee reviewed content to ensure there's no bias. CDC did not accept commercial support for this activity. CDC, our planners, presenters, and their spouses or partners disclose they have no financial interests or other relationships with the manufacturers of commercial products, suppliers of commercial services, or commercial supporters. Before we begin today's presentation, Laura Morrell with CDC's One Health Office will share some news and updates.
Thanks, Helen. Hello, everyone. Thanks for joining us for today's ZOHU call, and welcome to all of our new participants. The ZOHU call audience now has over 14,500 subscribers representing professionals from government, non-governmental organizations, industry, and academia, as well as students. We appreciate your help spreading the word about the ZOHU call. Please continue to share the ZOHU call website link with your colleagues. The site includes links to call recordings, information on free continuing education for a variety of professions, and a link to subscribe to the ZOHU call email list. To begin today's call, I'd like to share highlights from the One Health news from CDC included in today's ZOHU call email reminder. CDC has temporarily suspended the importation of dogs from Egypt, and USDA is enhancing African swine fever surveillance efforts. Some new resources that are available include an updated dengue website from CDC, free CE that's available for CDC's new module to help clinicians recognize and diagnose Rocky Mountain spotted fever. New CDC guidance for swine influenza is available on Medscape, and a new poster on staying healthy around classroom pets is available on our Healthy Pets, Healthy People website. Some upcoming events of interest include a CDC webinar on June 13th about emerging issues in tick-borne diseases, and the American Society for Microbiology Conference in San Francisco from June 20th through the 24th. We've also shared some recent publications, including baseline data on antimicrobial use by beef, feedlots, and swine operations from USDA, and human Bartonellosis, an underappreciated public health problem, as well as the June EID journal having a zoonoses focus. And in the MMWR, we have hurricane-associated mold exposures among patients at risk for invasive mold infections after Hurricane Harvey, and notes from the field, live poultry shipment box sampling at feed stores as an indicator for human salmonella infections in Michigan. There are several new outbreak notices that have been released, including an outbreak of E. coli infections linked to flour, outbreaks of salmonella infections linked to backyard poultry, multi-state outbreak of gastrointestinal illnesses linked to oysters imported from Mexico, and 4 ongoing outbreaks have also been updated. A selected list of ongoing and past U.S. outbreaks of zoonotic diseases, as well as info on staying safe and healthy around animals, is available on CDC's Healthy Pets, Healthy People website. The complete CDC current outbreak list is available at cdc.gov/outbreaks. If you would like for us to share news from your organization, or if you want to suggest presentation topics or volunteer to present, please contact us at zohucall@cdc.gov. Again, thank you for supporting the ZOHU call and for joining us today. I'll now turn the call back over to Helen.
Thank you. Today's presentations will address one or more of the following objectives. Describe 2 key points from each presentation. Describe how a multi-sectoral One Health approach can be applied to the presentation topics. Identify an implication for animal and human health. identify a One Health approach strategy for prevention, detection, or response to public health threats, or identify 2 new resources from CDC partners. Questions for all presenters will be taken at the end of the call. Call 1-800-593-8936 and enter participant passcode 9611836. Press star 1 and give the operator your name and affiliation. Please name the presenter or topic at the beginning of each question. You'll find resources and links for all the presentations on our website and in today's ZOHU Call email reminder. Our first presentation, Chronic Wasting Disease Overview and Update, will be given by Brian Richards. Please begin when you're ready.
Good afternoon. My name is Brian Richards. I'm a wildlife biologist with the USGS National Wildlife Health Center in Madison, Wisconsin. Our science center conducts diagnostics and wildlife disease research and provides epidemiological and technical assistance for states, tribes, and federal partners. We are the only federal facility with focus on wildlife health solely for the benefits of wildlife. The center's been around since 1975, and we've been involved with a number of high-priority wildlife disease issues since that time. We've been involved with chronic wasting disease, or CWD, since 2002, immediately after it was detected in the state of Wisconsin. Today, I'll be giving you an extremely short primer and update on CWD in North America. This slide lists some of the characteristics of CWD. CWD is a transmissible spongiform encephalopathy, the same category of diseases which include Creutzfeldt-Jakob's disease in humans and bovine spongiform encephalopathy in cattle. The hallmarks of all TSCs include a long incubation period, progressive neurological degeneration, a relatively brief clinical phase followed by death. CWD and its cousin scrapie in sheep are unique among TSCs in that diseased animals shed infectious agent via bodily fluids and can directly infect other members of their species. Transmission can be via direct contact or indirect with a contaminated intermediate environmental fomite. Vertical transmission has been documented but likely plays a small role in nature. Incubation period is protracted, followed by a brief clinical phase. Infected animals shed infectious agent long before the onset of clinical disease, and shed prions can persist in the environment for years, likely decades. We tend to see higher infection rates in males and in older age classes of each sex. We can test for disease ante-mortem, but the majority of surveillance samples are collected from hunter-killed animals post-mortem. At this time, management tools are lacking, and there is little evidence of widespread genetic resistance. The combination of these characteristics makes CWD extremely challenging to manage in free-ranging populations. But despite these characteristics, it is regularly argued that CWD is not a serious disease and that we should not attempt aggressive management in free-ranging or captive cervid populations. In 2016, Drs. John Fisher and Mike Miller presented a paper at the North American Natural Resources Conference describing the first 50 years of our experience with CWD. In it, they posed 2 overarching reasons why we should be concerned about CWD. First, that CWD in the long term is very hard on cervid populations. The second is that we cannot rule out that CWD could become a zoonotic human health issue. I'm going to go through some data and science in support of these 2 arguments. First, trends and outcomes in deer themselves. And first we'll discuss geographic spread. The following series of maps depicts the change in documented distribution of CWD in North America over the last 2 decades. Here is the documented distribution prior to the year 2000 in both free-ranging and commercial captive herds, the latter denoted by dots. The maps will now progress quickly until the current time. It is important to note that these maps denote 2 processes. First, geographic spread, and also the discovery of epizootics that likely had been missed for protracted periods of time. That takes us up to the current time. CWD has now been documented in 26 states and 3 Canadian provinces in North America in free-ranging and or commercial captive facilities. On the captive side, CWD has been detected in approximately 112 facilities in the U.S. and 107 in Canada. CWD has also been documented in South Korea, Norway, Finland, and Sweden. Clearly, there's been a significant expansion in the geographic footprint of disease. Second, increasing prevalence has been documented in multiple jurisdictions. The list on this slide is incomplete, but shows those jurisdictions where strong long-term data sets are available. I'll focus in on one area in the state of Wisconsin. The red polygon depicts a 24 by 24 mile area in the CWD-affected zone. We see 2 trends in the data. First, the prevalence tends to be higher in older age classes than in younger animals. That is to be expected. The second trend is more troubling, that prevalence within age classes has increased exponentially over time. In this zone, mature males have approximately 50% prevalence, and 1 in 5 18-month-old animals are positive. From the population perspective, this level of disease penetrance is not sustainable. The final characteristic in cervids is documented population impacts due to disease. These have now been demonstrated in western herds of elk, mule deer, and white-tailed deer where herd performance, or lambda, is at or below 1.0. In Wisconsin, there's an ongoing study looking at herd performance. Early results suggest that annual survival is 2 to 3 times lower for CWD-positive animals. Of course, this is to be expected with an always fatal disease. When prevalence rises to high level, disease becomes an additive factor and population impacts are certain. That summarizes the trends we are observing in cervids. Now I will very briefly touch on potential human impacts. We could discuss this topic for hours. I've tried to summarize in a few relatively brief points. First, epidemiological studies have found no instances where CWD has crossed the species barrier into a human host. But some researchers have been able, in experimental models, to move CWD across that barrier. We would have to therefore conclude that the risk of transmission to humans is not zero. And some researchers had suggested that the species barrier is not static. Certainly we know, based on the trends observed in deer, that human exposure is increasing, likely exponentially. In response, human health experts have issued guidance. The WHO suggests that no portion of any animal with a TSE enter the food chain. Similarly, and out of an abundance of caution, the CDC suggests that hunters in areas where CWD is known to exist strongly consider having their deer or elk tested, and in the case of a positive result, that hunters should not consume that meat. Back to the original slide, reiterating why we might be concerned about CWD. And here I list some of the likely longer-term outcomes. There is no reason to believe that CWD will not continue to spread and grow. Population impacts will likely continue to occur in high prevalence locations. It is an open question whether these impacts will be felt on a more regional basis. In high prevalence herds, we are likely to shift to a younger population age structure, and that is of concern to some hunters. And according to Human Dimensions research, hunters or their families are likely to alter hunting behavior and avoid these high prevalence herds. So to summarize, the data and trends in cervids suggest that CWD is a significant wildlife health issue. Published science suggests we cannot rule out a human health risk. Management efforts have been largely unsuccessful. And stakeholders have been less than supportive of using intensive harvest and or culling to manage disease. Without better management tools, it's hard to see how we will be able to alter detrimental long-term disease outcomes. Thank you, and I'm available at this email address for any questions we do not have an opportunity to get to today.
All right, thank you very much for your presentation. Our next presentation, Field Identification Key and Guide for Bats in the United States of America, will be given by Clint N. Morgan. Please begin when you're ready.
Hi everyone, this is Clint Morgan. I'm a biologist with the Ecological Investigations Unit of the Poxvirus and Rabies Branch at CDC, and today I'll be introducing the Field Identification Key and Guide for Bats of the U.S. This was a project that we worked on throughout 2018, and the Field Identification Key and Guide was published in January of this year in the Occasional Papers, which is a publication put out by the Museum of Texas Tech University Natural Science Research Laboratory. And before I get too far, I'm going to go a little bit over the status of bat rabies in the U.S. In the U.S., bats are the most common cause of acquired human rabies infections. About 17 out of every 20 reported primary human rabies cases are bat infections. And in Washington State, during 2006 to 2017, rabies positivity for all bat species was 6%. And across the entire U.S., from 2010 to 2015, only 43% of rabies positive bats were identified down to species level, which isn't very much. And the rabies phylogeny is structured by the host species. So the rabies virus has kind of structured itself throughout multiple species, creating multiple rabies lineages as represented on the figure to the right. This is from Stryker et al. 2010. So we identified that there was a need by of public health professionals in the U.S. to identify bat species. It's limited, and we assume that this is because the tools available for bat identification in public health labs is not sufficient. If all the bats were ID'd to species level as well as virus characterization occurred, this will inform many different things, from detection of the host shift and translocation events to detection of epizootic or novel variants, changes in rabies reservoir epidemiology, as well as distribution and prevalence of rabies variants in specific bat species. Even like risk assessments and future testing considerations can be impacted by bat identification. And we've seen that proper bat identification can aid epi investigations during exposure events. On the right, we have a figure from Krishnayami et al. 2017, which demonstrates how species identification will help track down, or helped us track down the origin of a bat, which was found dead in a package of salad greens in Florida. And it was only once this bat was identified to subspecies level through genetic typing that we were able to identify the exposure location. And so there are different methods to bat identification. You can use genetic sequencing identification. This is good for achieving identifications with just pieces of specimens or damaged specimens or tissues. And it can assess subspecies and other detailed taxonomic relationships. Some of the cons to this is it's costly and time-consuming, and it requires specialized expertise and equipment, and it really can't be done in the field yet, although there are new technologies coming online that might be able to do that in the field. Another way is just morphological identification, looking at the actual external structures of the bat. Some of the pros are that it's relatively quick and inexpensive. You can do this identification in the field or lab with no specialized equipment necessary. Some of the cons would be it's typically requires a mostly intact carcass or specimen with all those external features preserved. And there is some specialized knowledge necessary to do this, and some of the physical characteristics may be variable, particularly color. And currently, there are many field guides available, but few bat identification keys. Here's a list of just some field guides that are very common that detail bat ecology, habitat reproduction, and other natural history aspects of the bat, as well as some key characteristics of these bats. However, there's no in-depth identification keys in these publications. There are some, bat identification keys available, particularly in specific regions in states such as bats of Texas or Colorado. There's even a key to the eastern U.S. bat. But to date, there's no, or until this publication, there's no identification key for the entire United States. And to create this, we used the Bats of Texas, which was authored by bat biologist Lauren Ammerman of Angelo State University, and she's a co-author on this publication. And we use that as the base because there's a key within that book that has 33 of the 51 bat species in the U.S. And we use measurements and specific characteristics from other publications such as Jacobs 1996, Reed 2006, Bats of Texas, Savoyas 2014, and Menzel 2002. And other literature was used, and we needed to have some illustrations in this book throughout the key, and so we collaborated with a bat biologist and illustrator, Krista Demery, from the Texas A&M Natural Resource Institute for those figures. We also included species distributions for all the species from the IUCN Red List, and we included all updated taxonomy, and with the inclusion of the previous taxonames also listed. And the bat species that are most commonly submitted to public health labs were bolded throughout this document. So how does this work? Well, there's a series of choices kind of leads the user to the correct bat name, ruling out all the others. And so it's just a series of paired statements or couplets laid out in a numerical sequence. This allows whoever the user is, whether it be a laboratorian or a biologist, to identify these bats solely on their external features and collection location. And so below is an excerpt of the beginning of the key, and you'll notice that we state that this key does not include non-native bat species or accidental migrants, with the exception of these two vampire bats, Diphyla equidata, the hairy-legged vampire bat, and Desmodus rotundus, the common vampire bat. And these were included due to their epizoological importance. We found it necessary to include these due to multiple factors. The fossil record shows vampire bats in the U.S. up until about 5,000 years ago, and there's been some recent modeling efforts that have predicted South Texas and South Florida as suitable habitat for the common vampire bat, Desmodus rotundus. And South Texas in particular, also has high densities of cattle and feral pigs, which are a main food source for the common vampire bat. So we recognize a kind of a future need to be able to identify these bats if they're ever discovered. And both of these bat species have distributions in northern Mexico. And there's been some recent evidence which shows that these species are moving further and further north. The common vampire bat, Desmodus rotundus, is already within 50 kilometers of the United States, and there's concerns about northward expansion of range due to climate change. And the hairy-legged vampire bat, we already have one record from South Texas in the 1950s. However, this is a disputed account sometimes. So will the user need special training to use this key? Well, hopefully no, outside of what we include in the introduction of the paper as far as measurements and kind of bat anatomy and some baseline understanding of how you go about identifying a bat. The point of this key is for the user to make accurate identifications of bats across the U.S. as quick and easy as possible. We try to avoid the overuse of anatomical jargon and some technical terms to kind of make it as simple and accurate as possible. So making this presentation, I realized that the best way to show off this key would be to do some bat identification exercises. So I have one bat to key out, and it'll kind of take us through the key, demonstrating the workflow and the illustrations. So here we have one bat discovered at an undisclosed area near Roswell, New Mexico, and it was submitted for testing. There's some measurements: ear length 27 millimeters, forearm length 54 millimeters, and mass is 12 grams. And so you have this bat in your hand, and you try to figure out, looking at this couplet, this is the first couplet, you try to see what features it has. So Does it have a nose leaf, which is a distinct upward, freely projecting triangular-shaped flap of skin at the end of the snout, similar to figure 4A? You look down, you see figure 4A, you see this projection of skin from the nose. Oh, or does it not have this nose leaf, such as figure 4B or 4C? Our bat clearly does not have a nose leaf, so the second option, that couplet, is correct. And on the far right, you'll see it says go to number 5. So in your key, you would just continue over to number 5, which asks if the tail is absent, or is there a narrow U-shaped band of membrane around the legs, or is the thumb length more than 10 millimeters, or is the tail present, and there's an absence of a deep groove around the nostrils and nose pad. Clearly, there is a tail present on this bat, and so you would continue on to number 7. So number 7 asks, is there prominent flaps on the chin, or does the tail protrude from the dorsal surface of the uropatagium? Or are there no notable flaps on the chin? Does the tail extend to or beyond the edge of the uropatagium? Well, again, looking at figure 5A, which shows the tail of this ghost face bat, our tail does not match this, and so the tail extends to the edge of the uropatagium, and so we go to 8. Does the tail extend conspicuously beyond the edge of the uropatagium, or does it not? Okay, yeah, that was just from the, okay. Yeah, so the tail does not extend beyond the edge of the uropatagium, and so then you would go to 12. Which asks, are the ears proportionally large relative to the body, more than 25 millimeters from notch to tip, or are they proportionally large relative to the body, less than 25? So you just take a measurement of the ear of the bat in your hand, and you see it's 27 millimeters, and that will tell you that we need to go to 13, which asks, is the color black with 3 large white spots on the back, or is the color variable but not black, and there's no white spots on the back? Clearly there are no white spots on the back, and it's not a black bat, so we continue on to 14, which asks, are there distinctive glands evident on either side of the nose, and does it have a tawny or yellow dorsal color? Or are there glands evident on either side of the nose, and the dorsal color is light brown to gray? Don't really see any of these glands on this bat, such as you would see in figure 8. And the color is more yellow than brown. So we would go to 15, which asks, are there flaps or lappets projecting forward from the base of the ears, as in figure 9 below? Or are the hairs yellow-gray at the tips and dark brown at the base? Doesn't look like that. I don't see any flaps projecting forward from the base of the ear, and the dorsal color is pale yellow. So this tells us that the bat is Androzois pallidus, or the pallid bat. And in this key, we also include some geographic information at the end of each identification. And that's it. So we've identified the bat, and so there's 2 parts to this key, to this manuscript. There's the key portion, and there's the guide portion. After you identify the bat using the key, it's best to then go to the guide portion to verify your identification, as well as look at the distribution to see if it's fitting. So this is just the segment of the key for this bat. You can see it has the species name and also some other, more specific descriptions of the color, any kind of specific characteristics, and the measurements. All right, and that's all I have for this presentation. Our hope is that this key can be used to increase the frequency and accuracy of bat identification by public health professionals. We also include some additional resources that are found in the appendix of this publication. And below is my contact info, as well as a couple links. One is for the PDF, which is freely available from the Texas Tech University Museum. and the other is just a link to our CDC bat rabies page for more information.
Hey, thank you so much for your presentation. Please stand by just a moment while we change presentations. Okay, thanks for standing by. Our final presentation, update on raccoon roundworm and Asian longhorned tick research and surveillance, is by Dr. Michael Yabsley. Please begin when you're ready.
All right, thank you very much for the invitation to speak to you guys today. I'm going to be talking about two very different systems, and so I will be speaking rather quickly. Those who know me know that's not a big deal. These, of course, will be posted later on, and so if you have any questions, you can refer back to the slides as well. So first I want to start talking about the raccoon roundworm, Felis ascaris procyonis. So the life cycle is shown here. Raccoons are the typical definitive host, and we care about this parasite mainly because when a variety of birds or mammals, including humans, ingest the eggs, they develop these larval migrants that can cause clinical disease and death. There's also potentially alternative definitive hosts, which includes domestic dogs, and so my talk today will focus primarily on these alternative hosts, the dog, and the little young raccoon shown there in the middle. That is the basis for the first study I'm going to talk about. So we were interested in looking at wildlife rehabilitators to see if they were exposed to Felis ascaris. This group of individuals is of particular interest to me because they have exposure to a huge number of young raccoons each year if they're one of those large facilities that take in a large number of raccoons. So we went out and we collected blood samples from 347 rehabilitators and gave them all a survey about how they handle the raccoons. And you can see here on this map that 24 or 7% of them had antibodies to Felis ascaris. The zip codes for the sampling is shown in yellow, and then the red dots are those that were positive for antibodies. And then all of these are laid over a map that shows the general prevalence of Felis ascaris in raccoons. And so when you see those really dark states and provinces, those are the areas that have really high prevalence rates in raccoons. So you can see that there's red dots scattered throughout the country, but they do focus in on those dark states and provinces. I can't go through all of the laundry list of questions that we asked these individuals, but just want to point out a couple relevant things. This was whether or not they use PPE or hand hygiene for when they're handling raccoon feces, which of course would represent a risk. And what I want to point out on the left-hand side there is that not everybody acknowledged wearing gloves when handling feces, and on the right, hand hygiene, not everybody washed their hands after handling feces. So when we looked at risk factors and statistical significance, the western region came out as being a risky area. Part of that, though, is probably because one of the centers in the far western part of the country had multiple positives within that single center. So obviously it was an issue with that center, not necessarily that it's a higher risk than, say, the northeastern United States, where we do see a number of positives in people and raccoons. Not surprisingly, the risk for people being positive for antibodies is greatest in those areas that have high prevalence rates in raccoons, and also for those individuals that do not always wash their hands after handling feces or raccoons. So that particular study involved collecting blood samples from people as well as filling out a survey. We wanted to do another survey that didn't require blood sampling, so we got a lot more people to actually step up and help us out here. But this second study is actually published in two different papers that I want to talk about the knowledge and attitudes and practices of wildlife rehabilitators in relation to the raccoon roundworm. So again, there was lots of data in there, and I'm not going to go through all of it, but I just want to touch on a couple of things. This is a series of questions that had correct or incorrect answers. The most likely to answer correct are at the top here, and just highlight a couple. People, for the most part, knew that this parasite was not contagious between people. They generally knew the route of transmission, and they knew, which is important, that animals with Bacillus ascaris infections could present with neurologic signs that would resemble rabies and would be rabies suspects. Jumping down, a number of people didn't realize that fresh feces weren't necessarily an infection risk, which isn't always a bad thing because there's lots of other things you can get from feces. This parasite does take about 10 to 14 days to become infectious, so there is a window of opportunity where you can sanitize and prevent transmission. A number of people did not know how the parasite was actually diagnosed in raccoons or even other hosts, and most importantly, they did not realize at the age that raccoons could begin passing eggs. They can pass eggs as young as 8 to 10 weeks, and that's important because the vast majority of raccoons that these individuals are taking into their centers are quite young. Most of them thought they had to be much older before they represented a risk for exposure to Bacillus ascaris. So when we took all of the questions, including the ones I didn't talk about, and we compared that to a variety of characteristics of that individual, some significant factors for answering more questions correct included those with experience as rehabilitators for a number of years or decades, those that were members of professional rehabilitation groups and they would go to these meetings and get continuing education, those that had higher education level, primarily veterinarians, and those that have actually dealt with Bacillus ascaris in their facility before. So all of those make sense and really boils down to the importance of mentorship and education of these rehabilitators. And that's quite important because a large number of rehabilitators do this on the side and don't necessarily get the experience or education because it costs money and time to go to these groups and meetings. I want to shift gears now to looking at some of the work we've done with dogs. So dogs can play a number of different roles as hosts for Bacillus ascaris. On the left-hand side there, you've got puppies. If puppies ingest infective eggs, they can actually develop larval migrans and get sick and die. The two right-hand sides represent what would happen if an adult dog ingests eggs. They can either become patently infected with adult worms in their intestines and pass eggs into the environment, or they potentially could just ingest raccoon feces and serve as a transport host with those eggs passing out in the feces. Either way, for those two, it would be an important risk for people because dogs would defecate near homes and potentially bring those eggs into areas where people could be exposed to them. So we worked with IDEXX Laboratories to get fecal float data for a 3-year period to look at the distribution and prevalence of Bacillus ascaris in dogs. And we got almost 9.5 million data points from across the United States. And you can see there that the prevalence rate was actually quite low, 0.005%, so only 504 positive dogs. But when you look at the distribution of that, which is yellow is 1 report of a dog and orange is more than 1 report at the county level, you can see many of those fall in line with where we would expect Bacillus ascaris to occur based on high prevalence in raccoon populations. And when you exclude all of those negative states around the country, the prevalence in dogs does increase at a local level, but it's still quite low. So that's good news. When you look across the different breeds, there were a number of different breed classes represented as being positive. Also a wide range of positives occurred based on age, but importantly, if you look at that top color, the light green, we did see infections in dogs that were less than 2 months of age, suggesting that there could be transplacental transmission of this parasite, resulting in very young puppies passing eggs. So these data suggest that dogs may not be a great host for this parasite, so we did an experimental study looking at dogs as well as raccoons as controls, and not raccoon dogs for those that know what that species is. It does exist. So we looked at a number of different things, simply infection success, as well as when they started passing eggs, how many eggs they passed, how long they passed eggs, and then we also wanted to evaluate a new commercial assay that's been developed by IDEXX. And that particular assay detects antigen in feces, not necessarily the egg itself. So it, for Toxicara, works very well because it can detect infections prior to eggs being passed in the feces. So it provides a pre-mortem test, pre-patent test, sorry. So for the raccoons, we had 2 different exposure routes, eggs as well as larvae from mice. And as you can see in this slide, most of the raccoons did become infected and became patent, and most of them developed very high eggs per gram of feces, so 9,000 or more within those. When we looked at the IDEX copra antigen test, all of those patent raccoons did test positive, and on the right-hand side you can see that there's a dip in the antigen detection, and that's the point at which we treated these raccoons and they cleared the infection. So the test accurately detected the lack of parasites once they were cleared after treatment. The dogs were a different story. We still had 12 dogs that were exposed by 2 different routes, but only 2 dogs became patent, and these dogs became patent several weeks after the raccoons did, so it was the delayed patency in them. The other important thing to note is that the number of eggs per gram of feces was quite a bit lower in both of these dogs, and then both of these dogs self-cleared, so they didn't even get to the point where we treated these dogs. When we look at the copra antigen test, again, it detected infections in both of those dogs, and it detected infections in the dogs prior to them being patent as well, so similar to the Toxicara data. So overall, these studies show that raccoons are much better hosts than dogs. It's to be expected. They are the natural host. Of note for the IDEX data, those were all dogs under veterinary care, so it is a well cared for population, so we're certainly interested in looking at unowned dogs or dogs that don't receive veterinary care to see if prevalence may be higher in those populations. But regardless, these data do show that dogs can become patently infected, and they can also pass eggs in the feces, which represent a risk. And excitingly, the commercially available copra antigen test detected those infections as well. So, shifting gears to Haemaphysalis longicornis, the longhorn tick. So for those who don't know, this is the Asian longhorn tick, or the cattle bush tick if you're from Australia. It's native to Southeast Asia, but it has become invasive in a number of countries, Australia, New Zealand, and some other Western Pacific islands as well. It is important to the agricultural industry in some of these areas because it can cause stress, reduce growth, and transmit pathogens of importance to cattle. This tick is quite unique in that it can reproduce parthenogenetically. So for introductions, you only need a single tick, just a female. And for each generation of ticks that are produced, they're all female, and so you're doubling the number of ticks produced per generation because it doesn't produce any males. Within its native range, as well as some of the introduced populations, it's known or suspected to transmit a number of important pathogens of importance to people as well as animals. This is a laundry list of those here. None of these have been associated with Haemaphysalis longicornis in the United States yet. And currently there are no data to suggest that H. longicornis populations in the U.S. are transmitting any pathogens yet, but that work is, of course, still ongoing. So cows are the primary host for this tick, but it does have a huge host range that includes a large number of mammals as well as bird hosts, so that's, of course, important for its ability to invade a new area and establish itself. So looking at the United States situation, back in late 2017 in New Jersey, there was a sheep that had never left the country that was infested with a large number of ticks. These ticks were sent off and identified as the Asian longhorn tick. So you can see a picture there of the ear of that ewe covered in ticks. And this was the first report of this tick in the United States outside of quarantine. And so we went up to that index site later that fall and sampled some deer, but did not find the tick. So we hoped that it would not overwinter, but we went back up in spring of 2018 and unfortunately detected the tick on deer, possums, and raccoons near that index site. So it had survived the winter and established. Things then quickly went crazy. In May of 2018, it was found in several other counties in New Jersey. And then folks at Rutgers, who made the original identification, did a statewide tick blitz and found it in a couple other counties in New Jersey. Then it was found in Virginia, and then it was found in West Virginia, and then Arkansas. So things went rapid during early 2018. Folks went back and started looking at archived specimens of Haemaphysalis that they may have, because we have a species here in native to North America called Haemaphysalis lepora palustris, which is the rabbit tick. And unfortunately, there were some misidentifications, and so there were archived ticks from deer from September 2017 in West Virginia that were the Asian longhorn tick, and then in North Carolina on an opossum back in 2017. And then all the way back to 2010, some ticks that were collected off of a deer in West Virginia were positive. So this tick wasn't just in the country from 2017 forward. It's been here since at least 2010. So we here at UGA have been doing wildlife surveillance for this tick through a variety of different mechanisms, active trapping and surveillance, passive sampling of animals admitted to wildlife rehabilitation centers, as well as sampling of animals through a variety of different surveys and research projects that we have ongoing, as well as active environmental sampling as well. So this map right here in blue shows states that currently have confirmed reports of Haemaphysalis, and unfortunately, after I submitted these slides, Tennessee became positive. So you pretty much can't produce a map without it going out of date within a week, it seems. So all of those states in blue, including Tennessee, have confirmed HL from one source or another. Those numbers that are inside of the states are the number of wild animals that we've sampled. So to date, we've looked at over 1,000 wild animals in 17 states, and we've found them in all of those bolded states right there. And importantly, for North Carolina, West Virginia, Pennsylvania, Maryland, and Kentucky, wildlife surveillance actually represented the first detection of the tick within those states. We found it on a wide range of wildlife species listed on the left-hand side. Most of all of those are mammals, but it does include a red-tailed hawk, which is a concern, of course, anytime a bird or a migratory animal becomes infested, they can move at greater distances. Work by USDA and a number of other groups have found this tick on a number of different domestic animals. Work we've been doing with Wildlife Services, USDA found it on a skunk, and then there have been several instances of people becoming infested as well. Work that we've done, as well as some recently published work from Staten Island, have been looking at small rodents, Peromyscus in particular, to see if they are infested with the tick, mainly because of concern that if those small rodents are infested, this tick may play a role in transmitting important pathogens like Borrelia burgdorferi or Anaplasma. But to date, we have not found the tick on those small rodents, which is good news. We've been working on developing molecular diagnostic assays that can be used to distinguish H. longicornis from native ticks from North America, as well as Haemaphysalis from other countries. So if we get in very small larvae or damaged ticks that can't be identified morphologically, we can actually identify them within a few hours using a RIF-LIP assay. We've been looking at haplotypes, so sequencing a couple genes from the mitochondrial region, and there are at least 3 different haplotypes in North America right now, which suggests that at least 3 different females were introduced to this country. And we, of course, are interested in the origin of these ticks, so we've been doing next-gen sequencing of the entire genome of ticks from North America, as well as acquiring ticks from a variety of native populations and introduced populations in Australia. And this data is extremely preliminary. We literally have ticks rolling in every week that we're moving through this system, but essentially what this shows is that there's a large amount of variability across the genome of ticks, both from North America and abroad. We maintain a website, and so we try to get data from others and keep this website up to date for other data, but for our data that we acquire from wildlife hosts, we update this every week or so. So you can access the website there, and you can sort by state or county or host and look at the reports that way. And with that, I will thank a huge number of people who are involved in these 2 studies and welcome any questions that you may have now, or you can email them to me at that email address in the middle later. Thank you.
Hey, thank you so much. At this time, we'd like to take questions for any of our presenters. Call 1-800-593-8936 and enter participant passcode 9611836. Press star 1 and give the operator your name and affiliation. Please name the presenter or topic at the beginning of your question. Ted, do we have any questions on the line?
I'm not showing any questions yet, but again, if you would like to ask a question, please press star 1 and state your name and affiliation. Thank you.
going to check back. Ted, do you have any questions?
I'm not showing any questions yet, but again, if you would like to ask a question, please press star 1.
Thanks.
There is one question in the queue from Chris Haddock from the Centers of Disease Control. Your line is now open.
Oh, hi. This is a question for Michael Yabsley. Michael, great talk. Thank you very much. I had a question regarding seroprevalence to Balis ascaris. I guess, how do you interpret that, exposure versus infection? What are the determinants that go into, whether or not somebody is going to actually develop disease, develop visceral larval migrans after exposure to Balis ascaris eggs?
Yeah, that's a great question. And inclusion criteria for this particular study was a healthy adult. So none of these individuals necessarily presented with any clinical signs suggestive of larval migrans. We expected that we would be detecting antibodies in individuals that don't have clinical signs, even though most people consider Balis ascaris to be quite pathogenic because we've seen all the case reports that are published. But these individuals are all adults. They're being exposed to probably very low numbers of eggs accidentally, either by a lapse in protocol for cleaning cages or handling feces or something like that, versus the young children that tend to ingest a huge number of eggs at a single time. For purposes of this study, we called the individuals and let them know that they were positive, and if they had questions, and referred them to their doctor, because, of course, we weren't able to give any medical advice to these individuals. But for the vast majority of them, they, were perfectly healthy. They didn't seem overly concerned about it.
Great. Thanks very much.
I'm showing no further questions at this time.
Okay, we'll just wait one more moment for any last questions before we wrap up. Okay, just check one more time. Any questions? Last call.
There is a question in the queue from Kelly. Your line is open.
Perfect. Mike, can you hear me okay?
Yes.
Hey, Mike, thanks so much. This question is for Dr. Yabsley. Thank you so much for this presentation. I just have a quick question about the difference in life stages, the Balis infections that we see in puppies versus dogs. Or versus adults. Is there a reason that you can think of why we won't see larval migrans in the adults? Is that a change in the immune system, or what's the justification for that?
We see a similar phenomenon with young raccoons, and so young raccoons become infected at quite a young age, and then over the course of that first year, the worm burden tends to decrease, and then there's a typical purge of worms through the overwintering period. And so it's presumed that there's some level of immunity that develops to those parasites. And then when you reach adulthood for the raccoons, prevalence tends to be much lower than for the juveniles. Worm burdens tend to be lower as well as egg output. So there could be multiple things going on, but we believe it's probably something to do with an immune reaction that occurs later on that reduces the worm burden, and then possibly lack of an immune reaction in the very young animal that prevents those parasites from migrating at the beginning.
Okay, is there one more question before we end up?
I'm showing no further questions at this time.
Okay, great. Thanks, Ted. So thanks to everyone who did ask questions, and thanks again to all of today's speakers for their excellent presentations. Instructions for receiving continuing education are available at cdc.gov/onehealth/zohu/continuing education. The course access code is OneHealth2019. To receive free CE for today's webcast, complete the evaluation at cdc.gov/tceonline by July 8, 2019. A web-on-demand recording of today's call will be posted online by July 9, 2019 at cdc.gov/onehealth/2019/june.html. Our next call will take place in 2 months on Wednesday, August the 7th at 2 p.m. Eastern Time. Please send suggestions and questions to zohucall@cdc.gov. For more information and to subscribe to our email newsletter, please visit cdc.gov/onehealth/zohu. Thank you again for your participation. This ends today's call.